Quantitative structure-activity relationship models are regression or classification models used in the chemical and biological sciences and engineering. Like other regression models, QSAR regression models relate a set of predictor variables to the potency of the response variable, while classification QSAR models relate the predictor variables to a categorical value of the response variable. In QSAR modeling, the predictors consist of physicochemical properties or theoretical molecular descriptors of chemicals. The QSAR response variable could be a biological activity of the chemicals. QSAR models first summarize a supposed relationship between chemical structures and biological activity in a dataset of chemicals. Second, QSAR models predict the activities of new chemicals. Related terms include quantitative structure-property relationships when a chemical property is modeled as the response variable. As an example, biological activity can be expressed quantitatively as the concentration of a substance required to give a certain biological response. Additionally, when physiochemical properties or structures are expressed by numbers, one can find a mathematical relationship, or quantitative structure-activity relationship, between the two. The mathematical expression, if carefully validated, can then be used to predict the modeled response of other chemical structures. A QSAR has the form of a mathematical model. Activity equals F plus error. The error includes model error and observational variability, that is, the variability in observations even on a correct model. SAR and the SAR paradox. The basic assumption for all molecule-based hypotheses is that similar molecules have similar activities. This principle is also called structure-activity relationship. The underlying problem is therefore how to define a small difference on a molecular level, since each kind of activity, e.g., reaction ability, biotransformation ability, solubility, target activity, and so on, might depend on another difference. Good examples were given in the bioisosterism reviews by Pattini, Lavoie and Brown. In general, one is more interested in finding strong trends. Created hypotheses usually rely on a finite number of chemical data. Thus, the induction principle should be respected to avoid overfitted hypotheses and deriving overfitted and useless interpretations on structural, molecular data. The SAR paradox refers to the fact that it is not the case that all similar molecules have similar activities, types, fragment-based analogously. The partition coefficient, a measurement of differential solubility in itself a component of QSAR predictions, can be predicted either by atomic methods or by chemical fragment methods. It has been shown that the log P of compound can be determined by the sum of its fragments. Fragment-based methods are generally accepted as better predictors than atomic-based methods. Fragmentary values have been determined statistically, based on empirical data for known log P values. This method gives mixed results and is generally not trusted to have accuracy of more than plus or minus 0.1 units. Group or fragment-based QSAR is also known as GQSAR. GQSAR allows flexibility to study various molecular fragments of interest in relation to the variation in biological response. The molecular fragments could be substituents at various substitution sites in congeneric set of molecules or could be on the basis of predefined chemical rules in case of non-congeneric sets. GQSAR also considers cross-terms fragment descriptors, which could be helpful in identification of key fragment interactions in determining variation of activity. Lead discovery using fragnomics is an emerging paradigm. In this context, FBQSAR proves to be a promising strategy for fragment library design and in fragment-to-lead identification endeavors. An advanced approach on fragment or group-based QSAR based on the concept of pharmacophore similarity is developed. This method, pharmacophore similarity-based QSAR, uses topological pharmacophoric descriptors to develop QSAR models. This activity prediction may assist the contribution of certain pharmacophore features encoded by respective fragments toward activity improvement, and/or detrimental effects. 3D QSAR 3D QSAR refers to the application of force field calculations requiring three-dimensional structures, e.g., based on protein crystallography or molecule superimposition. It uses computed potentials, e.g., the Leonard-Jones potential, rather than experimental constants, and is concerned with the overall molecule rather than a single substituent. It examines the steric fields, the hydrophobic regions, and the electrostatic fields. The created data space is then usually reduced by a following feature extraction. The following learning method can be any of the already mentioned machine learning methods, e.g., support vector machines. An alternative approach uses multiple instance learning by encoding molecules as sets of data instances, each of which represents a possible molecular conformation. A label or response is assigned to each set corresponding to the activity of the molecule, which is assumed to be determined by at least one instance in the set. On June 18, 2011, the comparative molecular field analysis patent has dropped any restriction on the use of grid and partial least squares technologies, and the Rome Center for Molecular Design team has opened a 3D QSAR web server based on the 3D QuarterGrid R engine. GOLPE stands for Generating Optimal Linear Error Estimations. 3D QuarterGrid R covers all the main features of CumFA and Grid GOLPE, with implementation by multi-probe, multi-region variable selection that improves the simplification of interpretation of the 3D QSAR map. The methodology is based on the integration of the molecular interaction fields as calculated by AutoGrid and the R statistical environment that can be easily coupled with many free graphical molecular interfaces such as UCSF Chimera, Autodoc Tools, JMOL, and others. Chemical descriptor based in this approach, descriptors quantifying various electronic, geometric, or steric properties of a molecule are computed and used to develop a QSAR. This approach is different from the fragment approach in that the descriptors are computed for the system as whole rather than from the properties of individual fragments. This approach is different from the 3D QSAR approach in that the descriptors are computed from scalar quantities rather than from 3D fields. An example of this approach is the QSARs developed for olefin polymerization by half-sandwich compounds modeling. In the literature it can be often found that chemists have a preference for partial least squares methods, since it applies the feature extraction and induction in one step. Data mining approach computer SAR models typically calculate a relatively large number of features. Because those lack structural interpretation ability, the pre-processing steps face a feature selection problem. Feature selection can be accomplished by visual inspection, by data mining, or by molecule mining. A typical data mining-based prediction uses e.g., support vector machines, decision trees, neural networks for inducing a predictive learning model. Molecule mining approaches, a special case of structured data mining approaches, apply a similarity matrix-based prediction or an automatic fragmentation scheme into molecular substructures. Furthermore, there exist also approaches using maximum common subgraph searches or graph kernels. Matched molecular pair analysis, typically QSAR models derived from nonlinear machine learning, is seen as a black box, which fails to guide medicinal chemists. Recently there is a relatively new concept of matched molecular pair analysis or prediction-driven MMPA, which is coupled with QSAR model in order to identify activity cliffs. Evaluation of the quality of QSAR models. QSAR modeling produces predictive models derived from application of statistical tools correlating biological activity or physicochemical properties in QSPR models of chemicals with descriptors representative of molecular structure or properties. QSARs are being applied in many disciplines, for example, risk assessment, toxicity prediction, and regulatory decisions in addition to drug discovery and lead optimization. Obtaining a good quality QSAR model depends on many factors, such as the quality of input data, the choice of descriptors and statistical methods for modeling and for validation. Any QSAR modeling should ultimately lead to statistically robust and predictive models capable of making accurate and reliable predictions of the modeled response of new compounds. For validation of QSAR models, usually various strategies are adopted. Internal validation or cross-validation. The less data extraction perturb the original model. External validation by splitting the available data set into training set for model development and prediction set for model predictivity check. Blind external validation by application of model on new external data and data randomization or Y-scrambling for verifying the absence of chance correlation between the response and the modeling descriptors. The success of any QSAR model depends on accuracy of the input data, selection of appropriate descriptors and statistical tools, and most importantly validation of the developed model. Validation is the process by which the reliability and relevance of a procedure are established for a specific purpose. For QSAR models, validation must be mainly for robustness, prediction performances and applicability domain of the models. Some validation methodologies can be problematic. For example, leave-one-out cross-validation generally leads to an overestimation of predictive capacity. Even with external validation, it is difficult to determine whether the selection of training and test sets was manipulated to maximize the predictive capacity of the model being published. Different aspects of validation of QSAR models that need attention includes methods of selection of training set compounds, setting training set size and impact of variable selection for training set models for determining the quality of prediction, development of novel validation parameters for judging quality of QSAR models is also important. Application chemical. One of the first historical QSAR applications was to predict boiling points. It is well known, for instance, that within a particular family of chemical compounds, especially of organic chemistry, that there are strong correlations between structure and observed properties. A simple example is the relationship between the number of carbons in alkanes and the boiling points. There is a clear trend in the increase of boiling point with an increase in the number carbons, and this serves as a means for predicting the boiling points of higher alkanes. A still very interesting application is the Hammett equation, Taft equation and pKa prediction methods. The biological activity of molecules is usually measured in assays to establish the level of inhibition of particular signal transduction, or metabolic pathways. Drug discovery often involves the use of QSAR to identify chemical structures that could have good inhibitory effects on specific targets and have low toxicity. Of special interest is the prediction of partition coefficient log P, which is an important measure used in identifying drug likeness, according to Lipinski's rule of 5. While many quantitative structure-activity relationship analyses involve the interactions of a family of molecules with an enzyme or receptor binding site, QSAR can also be used to study the interactions between the structural domains of proteins. Protein-protein interactions can be quantitatively analyzed for structural variations resulted from site-directed mutagenesis. It is part of the machine learning method to reduce the risk for a SAR paradox, especially taking into account that only a finite amount of data is available. In general, all QSAR problems can be divided into coding and learning applications. Some models have been used for risk management. QSARs are suggested by regulatory authorities. In the European Union, QSARs are suggested by the REACH regulation, where REACH abbreviates registration, evaluation, authorization and restriction of chemicals. The chemical descriptor space whose convex hull is generated by a particular training set of chemicals is called the training set's applicability domain. Prediction of properties of novel chemicals that are located outside the applicability domain uses extrapolation, and so is less reliable than prediction within the applicability domain. The assessment of the reliability of QSAR predictions remains a research topic. The QSAR equations can be used to predict biological activities of newer molecules before their synthesis. When a chemical property is modeled as the response variable. As an example, biological activity can be expressed quantitatively as the concentration of a substance required to give a certain biological response. Additionally, when physiochemical properties or structures are expressed by numbers, one can find classification QSAR models relate the predictor variables to a categorical value of the response variable. In QSAR modeling, the predictors consist of physicochemical properties or theoretical molecular descriptors of chemicals. The QSAR response variable could be a biological activity of the chemicals. QSAR models first summarize a supposed relationship between chemical structures and biological activity in a dataset of chemicals. Second, QSAR models predict the activities of new chemicals. Related terms include quantitative structure-property relation and a mathematical relationship, or quantitative structure-activity relationship, between the two. The mathematical expression, if carefully validated, can then be used to predict the modeled response of other chemical structures. A QSAR has the form of a mathematical model. Quantitative structure-activity relationship models are regression or classification models used in the chemical and biological sciences and engineering. Like other regression models, QSAR regression models relate a set of predictor variables to the potency of the response variable, while